In this video, we're diving deep into the fascinating world of 3D tissue chips, a revolutionary technology that NASA has developed to simulate human organs for medical research. These chips, about the size of a credit card, mimic complex organ systems like the heart, lungs, and kidneys. By using them, NASA is tackling two major challenges: understanding the effects of space travel on astronauts' health, such as bone loss, muscle atrophy, and immune system changes, and advancing personalized medicine on Earth. Thanks to 3D tissue chips, researchers can now study human biology in unprecedented ways. These miniature avatars enable the testing of new treatments and drugs in conditions that closely resemble those found in the human body. This technology is also poised to revolutionize drug testing, providing safer, faster, and more effective options for patients, as it eliminates some of the traditional hurdles in clinical trials. In addition, NASA has partnered with other agencies to extend the longevity of these chips to 6 months, giving researchers even more time to study the long-term effects of space travel. As these miniature avatars continue to evolve, they could hold the key to unlocking new healthcare breakthroughs, both for astronauts exploring the vastness of space and for individuals on Earth. In the early 2010s, NASA embarked on a pioneering journey to develop 3D tissue chips, miniature devices designed to replicate human organs. These chips, about the size of a USB drive, were envisioned to simulate complex biological systems, enabling researchers to study human health in unprecedented ways. Recognizing the potential of this technology, NASA sought collaboration with other agencies. In 2016, the National Institutes of Health, NIH, through its National Center for Advancing Translational Sciences, NCATS, joined forces with NASA to launch the Tissue Chips in Space initiative. This partnership aimed to send tissue chips to the International Space Station, ISS, to observe how microgravity affects human cells. By May 2019, the first set of tissue chips modeling human organs like the lung and bone marrow embarked on their maiden voyage to the ISS. These chips provided invaluable data on cellular responses in space, paving the way for future research. In 2021, the collaboration expanded to include the Biomedical Advanced Research and Development Authority, BARDA, and the Food and Drug Administration, FDA. Together, these agencies selected 8 research projects to enhance the longevity of 3D tissue chips, aiming for a minimum of 6 months of functionality. This initiative sought to model acute and chronic exposures, such as drugs, radiation, and microgravity, to better understand their effects on human health. As of December 2021, tissue chips modeling the kidney and blood-brain barrier embarked on their second missions to the ISS, continuing the quest to understand human biology in space. Through these collaborative efforts, 3D tissue chips have evolved from a visionary concept to a powerful tool in biomedical research. The combined expertise of NASA, NIH, BARDA, and FDA has propelled this technology forward, offering new insights into human health both on Earth and in space. "3D tissue chips, also known as microphysiological systems, MPS, are small engineered devices that replicate the structure and function of human organs. Typically about the size of a USB drive, these chips contain living human cells arranged in a 3-dimensional configuration, closely mimicking the natural environment of tissues and organs. This design allows researchers to study complex biological processes in a controlled setting. The primary purpose of 3D tissue chips is to model human organs such as the heart, lungs, liver, and kidneys. By incorporating various cell types and recreating the microenvironment of these organs, the chips enable the simulation of physiological conditions. This capability is particularly valuable for testing how cells respond to different environmental factors, including radiation and microgravity, as well as various treatments like medications and chemotherapy. In essence, 3D tissue chips serve as sophisticated in vitro models that bridge the gap between traditional cell cultures and animal testing. They provide a more accurate representation of human organ function, offering insights into disease mechanisms, drug efficacy, and potential therapeutic interventions. This advancement holds significant promise for personalized medicine, allowing for treatments tailored to individual responses. By leveraging the unique properties of 3D tissue chips, researchers can conduct experiments that were previously challenging or impossible, leading to a deeper understanding of human biology and the development of more effective medical therapies. One remarkable example of a three-dimensional tissue chip is the lung bone marrow chip, developed through a collaboration between the University of Pennsylvania and the Children's Hospital of Philadelphia. This innovative chip replicates the complex interactions between the lungs and bone marrow, providing valuable insights into respiratory diseases and hematological conditions. The lung bone marrow chip consists of two interconnected microfluidic channels, one representing the lung environment and the other the bone marrow. This design allows researchers to observe how cells from the bone marrow respond to signals from the lung environment, simulating the body's natural processes. Such models are crucial for understanding diseases that involve both the respiratory and hematological systems, including certain cancers and immune disorders. By utilizing this three-dimensional tissue chip, scientists can test potential treatments and observe their effects in a controlled, human-like setting. This approach accelerates the development of therapies and reduces the reliance on animal models, aligning with the goals of personalized medicine. The lung bone marrow chip exemplifies the potential of three-dimensional tissue chips to bridge the gap between traditional cell cultures and animal testing, offering a more accurate representation of human organ interactions. As research progresses, such models are expected to play a pivotal role in advancing our understanding of complex diseases and developing effective, personalized treatments. Three-dimensional tissue chips are revolutionizing biomedical research by providing a platform to model human organs and study their responses to various conditions. Their significance spans both spaceflight and Earth applications, as well as the ability to model acute and chronic exposures over extended periods. In the unique environment of space, astronauts are exposed to microgravity and increased radiation, which can significantly impact human health. Three-dimensional tissue chips enable researchers to simulate these conditions and observe how human cells and tissues respond. This research is crucial for developing personalized medical countermeasures tailored to individual astronauts' sensitivities, ensuring their health during long-duration missions. Beyond space exploration, three-dimensional tissue chips have transformative potential for healthcare on Earth. They allow for more accurate modeling of human diseases, leading to better understanding and treatment development. Additionally, these chips can be used for high-throughput drug screening, accelerating the discovery of effective medications and reducing reliance on animal testing. Advancements in three-dimensional tissue chip technology have extended their functionality to model acute and chronic exposures over 6 months or longer. This capability is essential for studying the long-term effects of various stresses such as drugs, radiation, environmental hazards, infections, and microgravity. By maintaining tissue viability and physiological function for extended periods, researchers can gain deeper insights into chronic diseases and develop more effective treatments. Imagine predicting how a patient will respond to a treatment without ever administering it. With innovations like three-dimensional tissue chips and microphysiological systems, this is becoming a reality. These small devices, the size of a thumb, act as a crystal ball for biomedical research, offering insights into how treatments might affect a patient without risk. Currently, these chips can mimic human organs, but only live for about a month, limiting researchers to studying short-term effects. Longer-lived tissue chips could help fill in gaps by tracking the long-term impacts of treatments. This could lead to better predictions of how drugs behave over time, including potential side effects that aren't immediately apparent. Extending the lifespan of tissue chips to 6 months or more would transform biomedical research. It would allow for accurate long-term studies of drug safety, efficacy, and toxicity. These longer-lived chips could also drastically reduce the need for human and animal testing, offering a more ethical and efficient alternative. Additionally, they could help personalize medical treatments, both on Earth and for astronauts in space. Achieving this requires collaboration across various agencies. The FDA has been crucial in integrating these new testing methods into the review process. As FDA Chief Scientist Rear Admiral Denise Hinton explains, partnering with other agencies enables us to do things together that we can't accomplish alone, accelerating research and making it more cost-effective. The National Institutes of Health, through its National Center for Advancing Translational Sciences, has also played a key role in advancing these technologies. Dr. Joni Rutter, Acting Director of NCATS, emphasizes the importance of collaboration to speed up progress. Together, NASA, NIH, FDA, and other agencies are pushing the boundaries of biomedical research, improving healthcare on Earth, and advancing space exploration. These innovations promise a future where treatments can be personalized with incredible precision. The advancements in three-dimensional tissue chips are unlocking incredible potential for both space exploration and personalized medicine. By mimicking human organs, these chips allow researchers to predict how our bodies will respond to treatments, offering a glimpse into the future of healthcare. But here's a thought-provoking question for you. What if we could predict with precision how every individual will react to a treatment before they ever take it? Would this revolutionize medicine as we know it? Leave your thoughts in the comments below, and don't forget to like and subscribe for more fascinating updates on the future of science and technology.